Good morning everyone. Today we will discuss about the search of genetic material which was given by Frederick Griffith in 1928. In the previous video, as we discussed about the structure of the DNA, double helix model of the DNA, which was given by Watson and Crick. As you know, the DNA is the genetic material, but how it was discovered that the DNA is the genetic material in the living organisms? As you know, the nucleic acid was discovered nearly in 1869, and after the discovery of the nucleic acid, the discovery of the DNA as the genetic material took a long time to know whether the DNA is the genetic material. And so many scientists, they did the research about the component of the protein, RNA, DNA, and some they believe that the protein is the genetic material. But in 1928, Frederick Griffith, he conducted a series of the experiment, and finally his experiment was again restudied by, it was forwarded by some of the scientists, and finally it was known that DNA is the genetic material. So especially we will discuss about today that the experiment of the Griffith. The Griffith experiment is related to the transforming principle. You can say that the transforming principle or the transformation is the process in which one bacterium is going to be transformed into the another bacterium by receiving some of the genetic material. And that process of transformation was given by this scientist Griffith in 1928. So his experiment is transformation, or you can say transforming principle. So let us see how he conducted this experiment of transformation, what are the series of the experiments, and this experiment is very, very important for your examination. Generally this question is included in a 5 markers question. So just you try to understand the experiment of Griffith. Griffith conducted his experiment on a bacterium. The name of that bacteria was Streptococcus pneumoniae. You can see here he conducted the experiment Streptococcus pneumoniae, and the Streptococcus pneumoniae has actually two strains. One strain is S strain, and while other strain is R strain. S strain is also known as smooth strain, or you can say virulent strain, while R strain is known as rough strain, or you can write here a virulent strain. Griffith conducted his experiment on this bacterium Streptococcus pneumoniae. First of all, he cultured the strain of the Streptococcus this bacterium Streptococcus pneumoniae in the Petri dish, and he observed some of the bacteria. They look very smooth, their color was very shine, while some of the bacterium, some of the strain of this species Streptococcus pneumoniae, their surface was very rough. The bacterium, those who are having the smooth surface, he called this strain, that was the S strain. S stand for the smooth strain. And the bacterial colony in which the surface was very rough, he called this strain as the R strain. The S strain, as I told you, the surface is very smooth, while the R strain, the surface is very rough. But both the strains belong to this bacterium, and that is Streptococcus pneumoniae. While the surface of a S strain, this strain has the smooth surface, and here the surface is very rough. The smooth surface is due to the presence of the polysaccharide. Because of the slime layer, because of the polysaccharide, mucopolysaccharide, here the surface is very smooth, and in this case, in this strain of the Streptococcus, this polysaccharide coating on the outside, this polysaccharide coating is absent, and that's why this strain is said to be what? Avirulent, or you can say rough strain. So the smooth and rough is due to the presence of the polysaccharide, the mucopolysaccharide. If this layer is present, the mucopolysaccharide layer is present, the bacterium is said to be the S strain, and if this layer is absent, the strain is said to be what? R strain. So these two strains are there, S and R strain of this bacterium Streptococcus pneumoniae. The S strain or smooth strain is also known as virulent strain. What do you mean by virulent? What is virulency? Actually, virulency is the ability of any microorganism to cause disease. And if the strain is unable to cause disease, this is called a virulent strain. This strain, when S strain is going to be injected, when he injected S strain into mice died because of a disease, and that is called pneumonia. Let me to make here the diagram. Here, this is S strain. When Griffith injected S strain into the mice died. When he injected R strain of Streptococcus into the mice not dead, or you can say it was remain alive. S strain of the Streptococcus, as I told you, the Streptococcus pneumoniae is a pathogen, and Streptococcus pneumoniae is the causal organism, is the pathogen which causes pneumonia disease. In human being also, this Streptococcus is the cause of pneumonia disease. In mice also, Streptococcus pneumoniae can cause pneumonia disease, and because of the pneumonia, the mice can die. Here the two strains are there. S strain of the Streptococcus. When he injected S strain into the mice, mice died. Why mice died? Because of the pneumonia disease. As you know, the S strain is the virulent strain. It is the smooth strain. Because of the capsule, because of the mucopolysaccharide layer on the bacterium, it shows pathogenic in nature. It is virulent, and this strain, or you can say the virulent strain, can cause disease into the mice, and that's why the mice becomes dead. When Griffith injected R strain into the mice, mice was remain alive. It means mice not dead. Why? Because R strain don't have the capsule, don't have the mucopolysaccharide, as I told you. It is non-capsulated, you can say. The surface is rough. This is also known as avirulent strain. As you know, avirulent strain can't cause any type of disease. It can't cause the pneumonia disease in the mice, and that's why mice was remain alive, not dead. Now, in the third step, you can see here, Heatkilled S strain. When he injected into the mice not dead, means remain alive. Heat-killed virulent strain. What do you mean by heat-killed virulent strain? Heat-killed S strain. Here is also S strain, and here is also S strain. But what is the difference? Here S strain was heat killed. First of all, he took the S strain and then he heated, and because of the heating, the S strain in the culture media, they may be died. Their configuration, their component, they are going to be separated. And that's why when he injected heat-killed S strain into the mice was not dead, means they were remain alive. And how it was possible? When anything will be heated, definitely the bacteria, this strain, the S strain, or you can say the virulent strain, may be killed. And when it will be killed, when all the components, they are going to be separated, it will be no more pathogenic, it will be no more virulent, and that's why there will be no effect of this strain after heating, and mice may be alive, or you can say will remain alive. Now, in the next step, you can see here, heat-killed S strain plus R strain. He combined these two strain, heat-killed S strain and R strain both, and then he injected into the mice, and mice died. because of the pneumonia. Now, he thought that when the heat-killed S strain was subjected with the R strain, then the mice becomes dead. Then he came at the conclusion that it may be that some of the materials could have been transformed from heat-killed S strain to the R strain. Some of the genetic material, or you can say some of the, actually he used the term transforming principle, that some of the transforming principle may have been transferred from heat-killed S strain to the R strain, and later on R strain may be converted into the S strain, and that's why the the mice could have died because of the pneumonia. You can see here, it means the conclusion, or you can say the result, R strain is changed into S strain by receiving some of the transforming principle from heat-killed S strain, and this is called transformation. You can see here, this stage is very, very important. Heat-killed S strain, separately, when it was injected, the mice remain alive. But when the heat-killed S strain was mixed with the R strain, as you know, R strain is the A virulent strain. And when these two strains, both they are mixed together, and the mixture was injected into the mice died because of the pneumonia. How it was possible? Because R strain may have received some of the transforming principle, some of the materials, you can say, from the heat-killed strain, and later on R strain might have been changed into the S strain. Maybe that R strain may have again, there is the formation of the mucopolysaccharide outside, and finally it can transform into the S strain, and that's why the mice got dead. And this process is called what? Transformation. So what is transformation in this experiment? Transformation is the process of the change of R strain into the S strain by receiving some of the transforming principle from the heat-killed S strain, and because of the transformation, the mice finally got dead, or you can say becomes dead, and this is called transformation. The whole process is called transformation. Actually, in this step, when the S strain was heated, I don't know, bacterium, or you can say the bacteria is made up of mainly three components, that is RNA, DNA and proteins. When the S strain was heated, maybe that the protein, RNA, DNA, all they are going to be separated. Individually, now they are not able to transform, they are not able to cause the disease inside the mice, and that's why here in this step mice was remain alive. But when all these three components of the heat-killed virulent strain, that is proteins, RNA and DNA, they came into the contact of the R strain here in this step, maybe the R strain may have been transformed into the S strain by receiving some of the materials from the heat-killed S strain, and that's why the R strain changed into the S strain to cause the pneumonia disease into the mice, and that's why the mice died. But still, Griffith, he couldn't observe about the biochemical nature of the transforming principle. That actually what was the nature of the transforming principle, either it was RNA or DNA or protein, but definitely he said that something is coming from the heat-killed to the R strain, and that's why R is changed into the virulent strain, and he used the term transforming principle. But the nature of the transforming principle was later on studied by some of the scientists. I will teach you in the next lesson. And they studied about the biochemical nature of transforming principle, and finally it was known that that material was nothing, that transforming principle was DNA, and DNA is the genetic material. So in the separate heading, we will discuss about the nature of the biochemical nature of the transforming principle. But today, just you have to focus about the Griffith experiment. So once again, you can revise that what is Griffith experiment. The Griffith experiment means Frederick Griffith conducted the experiment of transformation on the bacterium, and the name of the bacteria was Streptococcus pneumoniae. As I told you, he studied, or you can say he conducted a series of the experiments on which bacteria? Streptococcus pneumoniae. And as I told you, the Streptococcus pneumoniae has how many strains? There are two strains, and these are S strain and R strain. S strain for smooth. When he cultured the Streptococcus pneumoniae, when he grown the Streptococcus pneumoniae into the petri dish, some of the colonies he observed, the colony was very shiny. It looks very shiny. And some of the colonies, they were very rough. That colonies which are very shiny, which looks very fine. That was because of the capsule which is made up of a mucopolysaccharide present on the above to the bacterial cell wall. So the colony having the the mucopolysaccharide layer, that colony is called S strain, smooth strain, and the colonies having the rough surface, or you can say which don't have the mucopolysaccharide layer above to the cell wall, that colonies, he used the term R strain. It means S and R strain both are the strain of the Streptococcus pneumoniae. Strain means a little bit, you can say the variety, a slight change is there in that particular species Streptococcus pneumoniae. So both they are same, S and R, both they are included into the Streptococcus pneumoniae bacteria. S strain of the Streptococcus is also known as virulent strain. Why it is said to be the virulent strain? Because it has tendency to cause disease into the mice. It can cause pneumonia disease. But R strain is known as avirulent strain. Why it is said to be the avirulent strain? Because R strain can't cause pneumonia disease into the mice, because there is no capsule, there is no mucopolysaccharide layer, as I told you. And in this flowchart, when the S strain he injected into the mice died because of the pneumonia disease, because this strain of the Streptococcus is virulent. It can cause disease into the mice, and that's why mice died because of the pneumonia disease. In the second step, when he injected R strain of the Streptococcus pneumoniae into the mice not dead, because this strain is avirulent, have no ability to cause disease because of no capsule, non-capsulated. This is also known as capsulated bacteria. This is non-capsulated bacteria. In the third step, what he did? He used the heat-killed S strain. Means this strain, S strain, was heated by him, and due to the heating process, the virulency is lost. The S strain, heat-killed S strain, will lose the virulency. And when this strain was injected into the mice not dead. Why mice not dead? Because it is going to be heated, and due to the heating, it can lose the virulency. It will be no more pathogenic, means can't cause the disease. Now, in the next step, what he did? Heat-killed S strain. This strain was subjected with the R strain, which is avirulent. And when these two strains were combined together and injected into the mice died. Now this is the step where you have to think that how the R strain is going to be transformed into the S strain. At this level, Griffith concluded that R strain has received some of the material from the heat-killed S strain, and maybe the R strain is changed into the S strain. And that material he used later on the term transforming principle. And that transforming principle can transform, can change the R strain to the S strain, and that's why the bacteria, that's why the mice becomes dead. So overall, he concluded the R strain is changed into the S strain by receiving some of the transforming principle from Heatkilled S strain, and this is called transformation. So I request you all to make this flowchart. You study once again through your NCERT book. All these steps are very important, and almost every year in the 5th markers some of the questions are related to this topic, search of the genetic material. So overall, the search of the genetic material, that DNA is the genetic material, was started from the experiment of Griffith, which initiated in 1929. Before, our scientists, they considered that maybe the protein, maybe the genetic material, and they were not arrived before his experiment that DNA could be the genetic material. So the DNA is the genetic material. We know, or the work started from his experiment only, from the Griffith experiment. And Griffith also, in this experiment, you can't say, Griffith also, finally, as I told you, he was unable to identify the biochemical nature of the transforming principle. Definitely he suggested that something is transferring from the Heatkilled strain to the R strain. Something, some materials you can say. And that material, he used the term transforming principle is the cause of the transformation, and that's why the R strain might have been changed into the S strain, which could be responsible for the pneumonia disease into the mice, and mice got finally dead. But the transforming principle may be DNA, maybe RNA, maybe protein. But later on, the 3 scientists I will discuss tomorrow in the next video, Abri, MacLeod, McCarthy, some of the scientists, they carry forward the experiment of Griffith transformation, and finally they identified that the transforming principle, the nature of the transforming principle, and that is DNA. So overall, the work started, or you can say the began, from the Griffith experiment. Almost, or you can say the work was done by Griffith, but later on the DNA was confirmed by some of other scientists. Tomorrow I will discuss the next heading, the biochemical nature of transforming principle. So that's all for today. Thanks everyone.